Hello, BISC130. This is the beginning of recorded lecture 3.1, starting off chapter 10 on cell reproduction. So again, we're getting bigger picture now. The first several chapters are about, molecules and organelles and stuff. Now we're talking about cells and how they make more of themselves. So not for the first time, and certainly not for the last time, we will be, when we talk about cells, comparing and contrasting prokaryotic cells and eukaryotic cells, because the way they do cell division is going to be different. So before we even talk about cell division, though, I want to give just a little bit of background about the genomes within these different types of cells. So let's start by talking about prokaryotes. The prokaryotic chromosome, their DNA, their genetic information, their genome. You can see here, it looks like a big old squiggly line. Remember, these things do not have a nucleus, so it's just floating around in the cytoplasm. If we wanted to isolate this genome, we would see that it's actually a circle. It is a single closed circle that just looks squiggled up when it's wrapped around like this. So the prokaryotic genome, and the term genome in fact is in the key terms. Key terms define genome as the total genetic information of a cell or organism. The prokaryotic genome is a single DNA macromolecule, and it is a closed circle. In contrast, eukaryotes, plants, animals, fungi, always more complicated, have not just one chromosome, but a bunch of individual chromosomes. The exact number depends on what species of eukaryote you're talking about. Eukaryotic chromosomes. This is human. So humans have 23 different chromosomes, but again, depends on the eukaryote exactly how many we're talking about. But yeah, importantly, it's plural. It's not just one like within prokaryotes. It's a bunch. So eukaryotic genome consists of several DNA macromolecules, several of these chromosomes. Humans, I'm not a fan of making you memorize a bunch of just specific numbers, but I mean, come on, we're humans, we should know this. Humans in particular have 23 different chromosomes, and this allows me to introduce a couple of other terms here. If we go back to this figure, we can see, yep, there's chromosome 1, chromosome 2, chromosome 3, chromosome 4, and so on, but we see 2 of each of these. And indeed, we have 2 different versions of chromosome number 1, 2 different versions of chromosome number 2, 2 different versions of chromosome number 3, and so on. The term for this is diploid. The key terms define diploid as a cell, nucleus, or organism containing 2 sets of chromosomes. However, it is worth noting, since we're just putting out terminology here, there's also the term haploid. So there are some cells, we'll talk about in the next chapter actually, and some organisms like some weird protists, some strange mosses. There are some organisms that are not diploid, but haploid. Haploid, reading from the key terms, a cell, nucleus, or organism containing 1 set of chromosomes. So we're actually going to use these terms several times in different chapters throughout this quarter, so you should be familiar with them now. Diploid means 2 copies of each chromosome. Haploid would mean 1 copy of each chromosome. The other important thing about eukaryotic chromosomes, which should be review from an earlier chapter, is that the DNA is not just a naked helix like it is within prokaryotic cells, but it is wrapped around these proteins called histones to form structures called nucleosomes. So this is a nucleosome. It is DNA wrapped around a histone. If we take a bunch of nucleosomes together, we form something called chromatin. So yep, we can see naked DNA, here's a nucleosome, here's a bunch of nucleosomes together. We call all this stuff chromatin. So in summary, again, these are not defined in the key terms. They're sort of defined in this sentence itself, but I underline the important terms in this statement. The DNA in the eukaryotic genome, the DNA double helix is wound around histone proteins to form many nucleosomes, which all collectively are referred to as chromatin. And again, none of this happened in prokaryotes. This is a eukaryote thing. The last thing to point out as far as eukaryotes go is our chromosomes are linear. They are not circles like prokaryotes. They are individual sausages, individual lines. There's one end, there's another end. Eukaryotic chromosomes are not circular, they are linear. Okay, so again, this is just some background about the chromosomes themselves. Now we can actually talk about the topic of this chapter, cell division. So, of course, we're going to talk about prokaryotic cell division and eukaryotic cell division because they are simpler. Let's start with prokaryotes. So the process of prokaryotic cell division is called binary fission. It's just the specific name for cell division going on in prokaryotes. So you hear binary fission, your brain should immediately understand this is a prokaryotic thing. So we could break this down across several steps. So step number 1 of binary fission is DNA replication. So I did say, just a few slides ago, that these prokaryotes have one chromosome. And that's true most of the time. But if this cell is getting ready to divide, to split in two, each of those resulting cells is going to have its own chromosome. So the first order of business here in binary fission is to duplicate that chromosome, to replicate that chromosome. The exact details of how this is done is going to be the topic of a later chapter. But for now, I'm just going to say step 1 of binary fission, DNA replication. Now, the other thing that each of these resulting cells is going to need is it's going to need cytoplasm. It's going to need space. They're going to need to be, the correct size. So next, the cell is going to get bigger. We call this elongation. You may notice, the 2 chromosomes here, these 2 circles, but we also notice some green dots here. Now, there are a lot of proteins involved in making all of binary fission happen. So I'm calling out, and the figure is calling out one of these proteins in specific, but I just want you to understand this is not the only protein doing a job here. It's just one of the most important ones. So these green dots are meant to represent something called FTSZ. A funky way of capitalizing it. It's how microbiologists like to name proteins. We'll see a couple more examples of this later in the quarter. But yes, during elongation, these FTSZ proteins start to move towards the center of the cell. We'll see why soon enough. So in summary, step number 2, cell elongation, and FTSZ protein moves to the center of the cell. Now, we know what's going to happen at the end here. By the time we're done with this, we're going to cut this cell in 2 and we're going to have 2 cells. Because we don't want to damage the chromosomes, if we're going to split this cell in half, we want the split to be down the middle, and we want these 2 chromosomes to be nowhere near the middle. We don't want them accidentally getting cut in half as the cell cleaves itself in 2. So during this next step, these chromosomes are going to be moved to opposite sides of the cell. And you can start to see this green FTSZ protein forming a ring. So again, this is a flat image, but remember, cells are 3-dimensional. This is a ring sort of pointing out at you and going back into the cell, specifically at the middle of the cell. So step 3, chromosomes move to opposite sides of the cell, and this FTSZ protein, many copies of it, form a ring there in the middle of the cell. Next, we're going to start building a wall. Remember, prokaryotic cells have a cell wall. So if you're going to split the cell in 2, you got to build a wall right down the middle. We call this wall a septum, and it's FTSZ that is directing the formation of this. Again, FTSZ isn't building this peptidic glycan polysaccharide. It's not building this septum. It's sort of this central coordinator. FTSZ is telling all these other enzymes and things that you're supposed to build this wall, this septum, here in the middle. Not right here, not right here, not across the middle like this, specifically in the center here. That's why, if I can go back a few slides, it was so important that the FTSZ goes to the center and forms a ring at the center, because we need to have this wall made at the center. So step 4, FTSZ protein directs the formation of this septum, a dividing cell wall in the middle or center of the cell. And finally, wall's in place. Now the plasma membranes just need to pinch off from one another, and we have what are called two daughter cells. A weird kind of terminology here. You have one parent becoming two daughters. There's no difference between these two, so it's not like one is the original parent and one is the offspring. Nope, one parent becomes two daughters. That's just the terminology. So step number 5, the cell pinches in two, forming two identical daughter cells. And here's a slide showing all these together if you want to look at it this way. So that was binary fission. That was the prokaryotic way of taking one cell, splitting it into two. Eukaryotes are, as always, going to be a bit more complicated. In eukaryotes, cell growth and cell division, so both parts of this getting bigger and then splitting in two, are part of a complex, what's called cell cycle. So I love this figure as a great overview of this entire process. So this is a circle that's just sort of ongoing, but if we want to hop in at the beginning of this circle, we would hop in, or right here, in what is called G1, capital G, subscript 1. So yeah, I'm double underlining eukaryotic cell cycle because these several stages are going to be underneath this. Starting with G1, the G stands for gap. So this is technically gap phase 1. What happens in gap phase 1? Well, boring background stuff, growth, and accumulation of resources. Again, the cell's got to get bigger if it's going to split in two, and as we'll see, it takes a lot of energy. It takes a lot of raw materials to go through the process of cell division, so just mustering your resources, getting everything ready is an important part of this. This chart is supposed to be drawn approximately to scale. So this slice of the pie looks like it's the biggest. That is intentional. This is the longest stage of the eukaryotic cell cycle. Again, just boring background stuff. I don't even have any pictures of cells in G1 because you can't really see what's happening here. They're just getting a little bit bigger and getting their resources together. Okay, after G1, we have S phase. The S stands for synthesis. Synthesis means building something or constructing something. What are you building? Well, DNA. So even though this is a lot more complicated than prokaryotic cell division, than binary fission, at the end of the day, the same basic stuff is going to happen in our cells. We're going to replicate our DNA, we're going to move the chromosomes to opposite sides, and we're going to split the cell in two. That's basically what's going to happen. So it just has different names to it. So yeah, S phase, synthesis, replicate the DNA, which does mean, if I can go back to this, we've got, as humans, 23 chromosomes, 2 copies of each, so that's a total of 46 chromosomes. After S phase, our cells are going to have, what's 46 times 2? 92 total chromosomes in these cells after they have completed this S phase. So yeah, every single one of these chromosomes is going to be replicated during S. It is also worth noting, as sort of a callback, you may remember this structure from inside of the cell, something called the centrosome, which contained centrioles, which contained microtubules. I introduced this in several chapters ago. This was an animal-only structure, and it is during this S phase, this synthesis phase, that specifically in animals, you get a replication of this centrosome. So we're going to have 2 of these in animals after we've finished S. So yeah, replication of centrosome contains centrioles, which contain microtubules. We'll see the job of this soon enough, but it's worth noting that it's synthesized, it's duplicated here in addition to all the chromosomes. Okay, so we had G1, S, next is G2. So gap phase 2. This is where we get replication of some organelles, stuff like mitochondria, chloroplast, lysosomes, peroxisomes, depending on exactly what type of cell we're dealing with. But yeah, some organelles are going to get replicated here, and just further growth and accumulation of resources. Just sort of another gap. Don't have a picture of a cell here because, yeah, most of this is not something that you can see. Now you may have noted that this arrow here that called this interphase, but it also called this interphase, but it also called this interphase. Interphase is kind of an umbrella term that includes these first three stages. So collectively, G1, S, and G2 are all referred to as interphase. Each of these three are part of interphase, which is just like the getting ready phase of the eukaryotic cell cycle. So yeah, this just refers to all three of these. Okay, next we have something called mitosis. And even though this is a slim slice of the pie here, there's actually a lot of complexity to what's going on during mitosis. So we're going to break down this slice of that pie itself into 5 different stages. So mitosis has several steps. These stages are going to be prophase, prometaphase, metaphase, anaphase, and telophase. And I will write those out as we come to those slides, but this sort of acronym is worth looking at because it's important to not get these things mixed up with one another, to know what happens in each of these phases. If you can remember PPMAT, you could remember the order of these phases. Prophase, prometaphase, metaphase, anaphase, telophase, that's the order. So, okay. Let's look at prophase. Zoom in on this. What happens during prophase is that these chromosomes, which are already wrapped around nucleosomes, they're already packed up, are going to start to pack even more tightly. The chromosomes are going to condense, and I like the term, become visibly distinct. So under normal circumstances in the cell, even under a microscope, you can't really see the individual sausages, but after prophase and during prophase, you start to be able to see, and again, this microscope image, it's difficult to see. That's why I kind of like the cartoon better. You can see the individual sausages. They're just gonna condense up as much as possible. The other thing that happens during prophase, and you can kind of see this here, the nucleus breaks down. So nucleus, obviously a very important organelle. It protects the DNA, but we know what's going to happen to these chromosomes. It's, like I said, essentially the same thing that happened in binary fission. We got to move them to opposite sides and split the cell in half. Well, we're not going to be able to do that if they are contained within a nucleus. So during prophase, we break down the nucleus, and while we're at it, we're going to break down the ER and Golgi as well. These structures are associated with the nucleus, so we're going to get rid of all these things. So prophase. Nucleus, Golgi, and ER break down. Chromosomes condense. They become visibly distinct. And something called the spindle apparatus forms. We can see it over here, this sort of blue spider-looking thing. I'm not going to say more about spindle apparatus now. We'll see what it does in just a second, which is prometaphase. So during prometaphase, here we've got the spindle apparatus, extends out its microtubules to attach to these chromosomes. So prometaphase, microtubules, part of that spindle apparatus, attached to sister chromatids. Okay, so here's where we need to zoom in a bit. So yeah, we can see that these are 2 chromosomes here, 2 sausages. Let's zoom in on these. So these 2 chromosomes are identical to one another. They are exactly the same. This is not talking about these 2 chromosomes. These 2 chromosomes are not exactly the same, and we'll get more into the implications of this in some of the later chapters. These 2 are not exactly the same. These 2 are not exactly the same. These 2 are not exactly the same. These 2 are exactly the same because we just had DNA replication during S phase. Every single one of those 46 chromosomes has an identical partner. These pairs of identical chromosomes are referred to as sister chromatids. don't know why they call it a chromatid and not a chromosome, but in this context, when they are partnered up like this, they are called sister chromatids. And in fact, there's a definition of this in the key terms. The key terms define sister chromatids as two connected identical copies of a chromosome. So, with that terminology out of the way, we can see microtubules, part of that spindle apparatus, attaching to the sister chromatids at a specific point called the kinetochore, spelled kinetochore, but this is the kinetochore. It is very important that these microtubules attach at the kinetochore because, again, we know what's going to happen eventually. They're going to get pulled apart from one another. They're going to move to opposite sides. And if we want these microtubules sort of tugging these things apart, we don't want some of the microtubules attached up here and down here and down here. That would rip the chromosome or the chromatid apart. We want to make sure that all the microtubules attach at these specific central locations, which are called kinetochores. So again, in summary, microtubules during prometaphase, microtubules attach to the sister chromatids at specific points called kinetochores. Okay, so they're going to get pulled apart from one another, but there's actually one more phase before we do that pulling apart. We have to make sure that everything is lined up nice and neat and orderly in the middle of the cell. Again, during prometaphase, these connections were made. The chromosomes were still kind of loosely just floating around where the nucleus once was. Metaphase is where everything lines up in the middle. Every single chromatid is positioned to go to one side or to the other side, two sides here. So this is metaphase. Sister chromatids align at the center of the cell, positioned to move to opposite sides of the cell. Now we finally do what we know is going to happen the whole time. They're pulled apart and go to those opposite sides. So this is anaphase. These sister chromatids are broken apart as these microtubules shorten, moving all these chromosomes either to one side or to the other side. And because every single one of these chromosome pairs they were identical copies, that means this set of chromosomes is exactly the same as this set of chromosomes. Again, just like binary fission, we are making two identical daughter cells by the time we're done with this whole thing. So anaphase, sister chromatids are split apart, and this spindle apparatus and its microtubules pulls these things to opposite sides of the cell. PPMAT, so after anaphase must be telophase. Here's where things start to go back to normal. The chromosomes are going to relax. They're going to decondense. You can see them starting to get kind of fuzzy and hazy. You won't be able to see the sausages anymore under a microscope. We also see during telophase the nucleus beginning to reform. The nucleus and the Golgi and ER. But it's happening X2. This is going to be a cell with two nuclei, each one with a full set of chromosomes, each one with its own ER and Golgi surrounding it. So telophase, chromosomes decondense, nucleus, Golgi, ER reform, X2. We got two of these. So importantly, and this trips a lot of students up, mitosis is not cell division. Look at this. We started with one cell, we end with one cell. Mitosis is not cell division. Mitosis is part of the eukaryotic cell cycle, and we're going to split this in two in just a second, but this process itself, mitosis, is dividing the nucleus. Mitosis is a process of nuclear division. We start off with one nucleus. It's got replicated chromosomes. By the time we're finished with mitosis, we have one cell, but there are two nuclei, each with a non-replicated or complete set of chromosomes. We've divided the nucleus. We have not divided the cell. So importantly, the stage that comes next is not part of mitosis. Again, mitosis is PPMAT. This next thing comes after mitosis, but it is not part of mitosis. The next stage after mitosis is called cytokinesis. And yeah, a cell pinches in two, and then you end up with two daughter cells. Not too much to this. In an animal cell, where there's no cell wall, this involves the formation of something called a cleavage furrow, which just has to pinch the membrane in two. In plant cells, which do have a cell wall, you have to build this wall down the middle, more similar to what we saw with bacteria. You build this cell plate down the middle, and then you pinch off the membranes. But either way, you're going to end up with two identical daughter cells as you finish cytokinesis. So, in summary, in cytokinesis, the cleavage furrow in animal cells, or the cell plate in plant cells, separates the cell into two identical daughter cells. And again, here's a good overview, but don't get confused. Prophase, prometaphase, metaphase, anaphase, telophase, these are mitosis. Cytokinesis, not part of mitosis. It comes after mitosis. Confusingly, we can lump these two together. So there is the term M phase. Just like interphase referred to G1S and G2, the term M phase collectively refers to mitosis and cytokinesis. So just another term that gets used describing the eukaryotic cell cycle. And again, I really like this slide as just a good visual overview for all of these steps and the order in which they occur. Now, there is actually one more stage that we could talk about. And if you're squinting hard at this and trying to search for it, it's actually not on this figure. There's one more stage that is technically not part of the cell cycle. That's, why it's not part of this figure. But it's a very, very important stage. This stage is called G0, capital G, subscript 0. And it's not part of the cell cycle. It's essentially an offshoot of the cell cycle. Cells in G0 finish cytokinesis, and then instead of going back to G1 and starting the whole thing again, will go off and be G0 instead. So again, this is kind of an offshoot, not technically part of the cell cycle. G0 is also known as resting phase. I really don't like that term, but it is the term that is used, and I'll explain why I don't like it. But G0, also known as resting phase, is not part of the cell cycle. It's an offshoot after cytokinesis, but before G1. So what's going on in G0? Well, the cells in G0 are not actively dividing. That's why it's not part of the cell cycle. If you're in G0, you're not getting ready to divide, you're not, accumulating resources, you're not preparing or whatever. Cells in G0 are doing nothing to do with the cell cycle. That's why it's called resting. But I don't like resting because it implies these cells are just doing nothing, which couldn't be further from the truth. Cells in G0 are just doing their job. Whatever the job of this cell is, they are doing it. Most of the cells in our body are in G0. Your skin cells, they're doing the job of, secreting oils and sweat and stuff like that. They're doing their job. They're not dividing. They're in G0. Your muscle cells, they're contracting when you tell them to do so. They're not dividing. They're doing their job. They're in G0. Cells in your liver, neurons in your brain, virtually all the cells in our body, they're not resting. They're not doing nothing. They're doing whatever their job is. They are in this G0 phase. So it's very important in that respect, in that most of our cells are not actively dividing. They're in this G0. So this is for cells doing their normal functions, not getting ready to divide or doing any of this other stuff. Very few cells in the body are actively going through this cell cycle. Okay, now let's talk about control and regulation. So this is obviously a very complicated process. None of this magically happens on its own. There is a lot of regulation that occurs here to make sure that it all happens without any mistakes. the cell cycle is regulated by internal factors. So the cell has sort of internal checkpoints like pause, like, am I ready to go forward? Should I continue? The cell checks itself in 3 main points. So there are 3 main cell cycle checkpoints, which are shown on this figure. So very similar to the last one, but this is showing the checkpoints here. The first of these is something called the G1 checkpoint. And wouldn't you know it, it happens in G1, near the end of G1. The G1 checkpoint is checking for energy and resources for DNA replication and for damage to DNA. So again, just think about this logically. If you're going to finish off G1, that means you're going to proceed into S phase, where you're going to replicate all the DNA. That takes a lot of energy to replicate DNA. That takes a lot of raw materials, a lot of nucleotides. You don't want to start this process if you're not ready to see it through. So the G1 checkpoint is going to check for this and is going to hold things up and wait, pause that cell in G1 until it's actually ready to go forward. Because that's the other thing about DNA replication. There's no rewind button on this. There's no pause button on this. You got to make sure you're ready before you leap. And it's also checking for damage to DNA. We'll talk more about damage and repair in later chapters. But yeah, you don't want to replicate damaged DNA. That's a recipe for disaster. So this checkpoint is also looking for damage and trying to fix it before you move to that very important step of replicating the DNA. Our next cell cycle checkpoint is called the G2 checkpoint, which, again, I like these names, is in G2. It's near the end of G2. And what is the G2 checkpoint checking for? Well, it's also checking for damage to DNA. And this sounds a little redundant with the G1 checkpoint, but at this point, it's been a long time since the last checkpoint. You replicated everything. You had some more growth. We're about to head into mitosis, and you do not want to have damaged DNA during mitosis. You want all your DNA to be in proper working order before we start moving these chromosomes around. So it's important to, once again, make sure that all this DNA is in proper working order, no damage. And if there is, make sure it's repaired before you have the green light to move from G2 to mitosis. And third is the M checkpoint, which is in mitosis, and you may think that the M stands for mitosis. Technically, the M stands for metaphase, because this M checkpoint happens during metaphase. What we're checking for here is we're checking to make sure that all of these connections are secure, to make sure that every single sister chromatid is attached to the microtubules, part of the spindle apparatus. We know what happens after metaphase. They're going to get pulled apart from one another. If any of these chromatids are not secured to microtubules, they will not get pulled apart, and that would be really bad. So we'll just secure the connections, double check all these connections in metaphase before they get pulled apart in anaphase. No take backsies once you start going through this process. So our third checkpoint is called the M checkpoint. M stands for metaphase. It checks that all sister chromatids are properly connected to the spindle apparatus. Now, these 3 were internal checkpoints, where the cell checking its own, status of its, proteins and its DNA and its energy levels and stuff like that. The cell cycle is also regulated by external factors. We got 40 trillion cells in our body, for example. They need to play nice with one another. They need to make sure that they only divide when they need to divide. So part of these external regulation is that cells need permission from surrounding cells to divide. We don't want our cells just dividing because they can or because they have enough energy to. We only want our cells to divide when we need them to divide, and we're going to send external factors. Hey, this goes back to the last chapter. this horrid figure again. We're going to send ligands that bind to these receptors. A big part of this gigantic signaling pathway is to control cell proliferation, to tell cells when they need to divide and when they don't need to divide. So this external regulation is incredibly important in maintaining normal cell number and normal cell density. We only need so many cells. We only want to divide if there's actually a need for that division. So cells are checking themselves internally. Cells are also being regulated by factors from the outside of the cell, from ligands binding to their receptors. And none of this regulation magically happens on its own. There are tons of proteins involved in signaling pathways, obviously, and there are tons of proteins involved in all these internal checkpoints as well. This is another one of those figures that you don't need to memorize or sweat the details on. I just like showing something that really illustrates the full complexity of everything that's going on here, that there are tons of proteins involved in all of these checkpoint regulation things or whatever. Doesn't happen on its own. Many proteins involved in regulating the cell cycle. Okay, we've got one more section on this chapter, but I don't think I can finish it in the normal amount of time that I have. So this is going to be the end of recorded lecture 3.1. I'll finish up cell reproduction in the next recorded lecture. But yeah, this is the end of what I got for 3.1.